One of the keys to a successful hydroponic garden is having a solid nutrient management plan. Plants grown hydroponically are much more sensitive to the nutrients you give them simply because they don't have soil to buffer any mistakes you may make. In soil, the plants await rainfall or irrigation to dissolve the nutrients and any other added fertilizer present in the soil so that the roots can search out and absorb the essential nutrients. In hydroponics, the plant's roots are constantly provided with water, oxygen, and nutrients, continuously making the job for the plant roots easy. However, as you are taking on nature's role, you must provide the right nutrients in the right amounts, with the right pH, and at the right time in the plant's growth cycle. The challenge is to keep up with the plant's needs to avoid damage from excesses and deficiencies, extreme pH and temperature, and lack of oxygen. The advantage of hydroponics is the great control it offers over health and quality of the plants. Here we will learn about the components and mechanisms of nutrient management required for successful hydroponic growing. Since hydroponics farming is soilless, the quality of its most basic component, water, is of primary importance. Well water contains dissolved elements from the ground from which it is pumped. These elements can help or hinder plants in a hydroponic system through nutrient excesses, deficiencies, or antagonism. City or municipal water, whether sourced from wells, rivers, or reservoirs, has similar dissolved elements. In addition to a filtration process, certain chemicals are added, such as chlorine, to kill bacteria. Other chemicals are added to raise the pH of the water to 7.5 or greater to prevent corrosion of the water pipes. And lastly, oxygen scavengers are added to the water to remove dissolved oxygen, again to prevent corrosion of the water pipes. All of these can greatly affect the quality and quantity of the nutrient blend and, in worst case, cause damage to the plants through interaction and antagonism. In case of well and municipal water sources, obtain a water analysis of your source water. Consult with a hydroponic nutrient company so they can assist in designing a modified nutrient plan that compensates for the elements present in the water. In our system, we process all our water through a reverse osmosis or RO water system. The RO system has pre-filters which remove the sediment and particulates, and a membrane which only allows the water molecule H2O to pass. This results in water with a 0.0 total dissolved solids and pH of 7.0, which can easily be adjusted as required. The first thing to understand about nutrients is the importance of each nutrient to the growth and health of the plant. Primary nutrients include nitrogen, phosphorus, and potassium. Large quantities are needed for the plants to thrive. Secondary nutrients are calcium, magnesium, and sulfur. They're needed in smaller quantities than the primary nutrients, but in much larger amounts than the micronutrients. Micronutrients include iron, molybdenum, boron, copper, manganese, sodium, zinc, nickel, chlorine, cobalt, aluminum, silicon, vanadium, and selenium. Environmental nutrients are oxygen, hydrogen, and carbon. The plants receive these from the atmosphere and from the water. Primary nutrient needs are supplied as part of a nitrogen, phosphorus, potassium fertilizer referred to as NPK. NPK blends are described using 3 numbers. For example, an NPK blend designated 10-10-10 means the mix is 10% nitrogen by weight, 10% phosphorus by weight, and 10% potassium by weight. The ratio of NPK depends on the plant you are growing and the stage of growth the plant is in. For example, nitrogen is a major component of several essential compounds such as chlorophyll and amino acids. It promotes structural and vegetative growth of stems and leaves. Phosphorus is a vital component of adenosine triphosphate, or ATP. ATP is used for energy, holding DNA and RNA together, and other major uses such as promotion of root and fruit growth. Potassium regulates the opening and closing of stomata during photosynthesis as well as promoting general plant growth. Stomata are a pair of specialized cells which regulate the gas exchange between the plant and the environment. Stomata lets in carbon dioxide and releases oxygen. In addition to that critical function, stomata releases excess water in a process called transpiration. Micronutrients are required in trace amounts and don't normally need to be added to a nutrient mix. Generally, the seed from which a plant germinates has enough of these nutrients to sustain a plant for its entire cycle. Normally, pH is used to control the specific nutrient availability to the plant roots. With hydroponically grown plants, the relationship between pH and micronutrients is a little different. The micronutrients are much more available to the plants from the nutrient solution, so it is more effective to regulate the actual concentration of micronutrients in your solution. Nevertheless, micronutrients are vital to the plant. Boron enhances the structural integrity of the cell walls. Chlorine is involved in the opening and closing of stomata during photosynthesis. Copper is involved in enzyme activation and metabolism of both carbohydrates and proteins. Iron is a major component of chloroplasts, which produce the chlorophyll. Manganese is required for chloroplast formation and nitrogen assimilation, but also important for pollen germination and root disease resistance. Molybdenum is crucial for production of two enzymes responsible for the uptake of nitrate from the soil and nitrogen from the atmosphere. Zinc is a key component of many enzymes and proteins. It is easy to be overwhelmed by the hundreds of nutrients and blends available. There is a wealth of information available on the internet regarding the specific nutrient requirements for the particular plants you would like to grow. Blending your own nutrients can also be daunting for newcomers to hydroponic growing. Several manufacturers have simplified the process. General Hydroponics, for example, has a 3-part system, which is a good place to start. FloraGrow is a 054 NPK blend that is mixed for early plant development. FloraMicro is a 501 NPK blend for rapid vegetative growth and includes micronutrients. FloraBloom is a 216 NPK for fruit development. The three are blended in various proportions depending on the stage of growth of your plant. Although it's a good place to start, it's not the best option for all plants. Lettuce, for example, likes high nitrogen and potassium, but low phosphorus. The MaxiGrow product is a 1-part 10-5-14 NPK blend. For fruiting plants after the growth phase, you change to a MaxiBloom, which is a 5-15-14 1-part blend specifically for that stage of growth. Liquid nutrients in concentrated form are easy to use and convenient. However, they cost more. You're paying for water. Larger hydroponic growers use a powdered nutrient and blends because it is much more cost effective, even though you're required to mix it together. In either case, it is important to keep exacting records of your nutrient blends and their effect on plant growth. Water, nutrient blend, and temperature of both root and leaf all affect growth. As nutrients are added to the base water, the total dissolved solids, or TDS, and electrical conductivity of the solution changes. If RO water is used, the initial TDS reading in parts per million, or the EC for that matter, is near zero. Adding nutrients in accordance with the manufacturer's instructions will raise the EC and the TDS. TDS and EC are measured using a meter. Some of these meters will display TDS in parts per million as well as EC. At this point, it's important to understand the difference between TDS and EC. EC measures the electrical conductivity of the solution. TDS measures the dissolved solids of the solution, all of them, even the ones that did not conduct electricity. However, a true TDS reading requires a lab test. A TDS meter measures the electrical conductivity of the solution and performs a calculation to determine the parts per million of the solution using one of two conversion factors. And herein lies the problem. Not all manufacturers use the same conversion factor. Thus, no two TDS meters read exactly the same. Better meters are able to do three-point calibration using known test solutions of a known conductivity. Meters need to be periodically recalibrated to ensure consistency. When initially filling a reservoir, make sure you record the number of gallons of water added. It becomes the basis of nutrient analysis throughout the nutrient life. Take and record an EC and TDS reading. Add the recommended blends of nutrients. Then take another reading and record. The difference between the initial reading and the second reading is the effect of the nutrients on the solution. Make sure these readings are recorded along with the amount of nutrients added to the system. These become your base readings. Next, adjust the pH of the solution to the recommended values and record. Temperature is another key to growth. Too cold and the seeds will not germinate, or the plants will grow slow and stunted. If the temperature is too high, then the same seed still will not germinate, and the plants can die from oxygen deficiency or just plain heat stress. Although the temperature of the grow space is important, we are discussing the temperature of the nutrient solution. As long as the plant roots are kept at their proper temperature, then the plants will grow. As discussed in another module, pH affects plant nutrient uptake. It can also be used to adjust the uptake of certain nutrients. For example, a high pH will inhibit iron uptake, even though the nutrient solution has sufficient iron. Nutrient antagonism and interactions. Nutrients are roughly taken up by the plants in roughly the same relative amounts that they are present in the nutrient solution. However, when one nutrient is in excess, it can be taken up at a higher amount at the expense of another nutrient. This is called nutrient antagonism. In this case, it is possible to have sufficient amount of nutrients and for the plant to be deficient in another. For example, excess potassium in the nutrient mix can cause antagonism with nitrogen, causing a nitrogen deficiency, even though the correct amount of nitrogen was added to the blend. Here is a chart which graphically shows the antagonism and synergism between various nutrients. The red lines indicate the antagonism between nutrients, and the arrows indicate the causal interaction. Copper reduces the availability of iron. Calcium reduces the availability of zinc. Certain combinations, such as phosphate and iron, reduce each other. The green lines indicate the synergism between nutrients. Molybdenum increases the availability of nitrogen, and phosphate and magnesium increase the availability of each other. Soluble salt damage can be caused by over-fertilization, poor water quality, and salts accumulating in the growing media over time. As water evaporates, soluble salts can accumulate in the aggregate media if they are not adequately leached out. Chemical-induced drought can occur when soluble salt levels in planting media are excessive. As a result, you will see wilting of plants despite adequate irrigation. Other symptoms include dark green foliage, dead and burned leaf margins, and root death. Soluble salt levels can be monitored and measured by tracking the electrical conductivity of the nutrient solution. The cure for this is to drain the reservoir and replace it with pure, pH-balanced water, and then to flush the entire system. It's important the pH match what's required by the plants. Test the returning water until the EC stabilizes. Common nutrient deficiencies. Nitrogen deficiency can be caused by underfertilization, nutrient imbalance, or excessive leaching. Typical first symptoms of nitrogen deficiency are light green foliage and overall stunting of plants. You can also see wilting and dead and/or yellow leaf margins. Measuring and monitoring the electrical conductivity of the nutrient solution can help prevent nitrogen deficiency. Adjust EC levels when they are low or high. Identify the source and correct it. It may mean adding more nitrogen to the solution. In addition, the cause could be an excessive antagonistic nutrient. In that case, drain the entire reservoir, flush with a pH-balanced clear water, and then add a fresh nutrient blend, ensuring the correct amount of each nutrient. Calcium deficiency can be caused by underfertilization, nutrient imbalance, or low pH. It is also related to moisture management, high temperature, and low airflow. As a mobile nutrient, calcium travels through the plant in water-conducting tissues. As fruit and leaves compete for water, a low humidity, high temperature condition can result in increased transpiration rates from the leaves. In this condition, the fruit develops a calcium deficiency. Deficiencies start out as brown leaf margins on new plant growth or on the bottom of the fruit. Correct the pH in the nutrient solution. Greenhouse airflow could also be low. Increase the horizontal airflow to between 0.3 and 1 meter per second to break the boundary layer on the leaves and to increase the transpiration rate. Make sure that the airflow is uniform over the plants. The most common cause of iron deficiency is high pH in the media as well as in the nutrient solution. Iron deficiency shows up as a yellowing between leaf veins, usually on new growth. Correct by adjusting the pH of the nutrient solution. Magnesium deficiency can also be caused by high pH in the media and an imbalanced nutrient solution. Indication of magnesium deficiency is yellowing between the leafy veins, similar to iron deficiency. However, magnesium deficiency shows up on the middle and lower leaves instead of on new growth, as in the case of iron. Correct by adjusting the pH. Boron has the narrowest range between deficiency and toxicity. It is easy to over-apply boron. If using city water or well water, boron can be present. An analysis will detect the amount present, and this needs to be accounted for in the nutrient blend. If using RO water, double-check the nutrient blend. The symptoms of boron toxicity are yellow and dead spots on the leaf margins. Stunted root growth is also a symptom. The cure is simple. Determine the source of the excess boron and correct it. Nutrient and nutrient management, although complex, can be achieved by following these rules. Water. Quality water is a great advantage. Poor water is a challenge. Nutrients. Use only the highest quality plant food, designed specifically for hydroponics. Low-grade plant foods and common fertilizers offer your plants poor and incomplete nutrition. They cause pH drift and sometimes contain impurities that can become toxic to hydroponic plants. Blending nutrients. When you mix fresh nutrients, always measure carefully and record your recipe. Data logging. Keep a daily log of parts per million, pH, EC, water usage, and temperature and humidity. Temperature and ventilation. Control your nutrient temperature. Too high a temperature affects dissolved oxygen and also increases root stress. Too low, this stunts plant growth. Observation. Observe your plants on a daily basis. Take note of their growth condition. Early detection of nutrient deficiencies can prevent the loss of the entire crop.